Well, this is the highlight of this morning for me because I've heard Dr. Christine Jones speak once before, and it was also clear to me then, following that presentation, the communication that goes on right beneath our very feet. Our keynote, delivered by an internationally renowned soil ecologist, researcher, speaker, and founder of Amazing Carbon, working with innovative farmers and graziers over several decades to implement and quantify land management practices that not only enhance biodiversity, nutrient cycling, carbon sequestration and productivity, but also water quality, as well as community and catchment health. Dr. Christine Jones has helped many of us, including me as I've mentioned, to understand the microscopic synergies occurring between fungi, bacteria and plants, creating the very life beneath our feet. Please welcome Dr. Christine Jones.
So that was a fantastic introduction, thank you very much. So I'm going to be talking about a regenerative grazing, soil and animal health, which we could talk about for a week. And I'm going to try and cram some information into about 40 minutes, so we have another 20 minutes for Q&A. Gilly's going to go like this when I get to 40 minutes so that I stop. If I don't get through all my slides this morning, I have got another presentation this afternoon and I'll just slot them into there so we won't miss out on the information. Because the thing about grazing, and as that kind of work that's happening with the mine site there and also happening in other places around the world, and things that we've become aware of over the last couple of years, is that grazing that improves soil health will also improve animal health. And well, in this case, now we're not talking about bioremediation, where we're actually talking about, grazing on a farm. If the animal health isn't improving, then there's something missing. So that's why I have changed the title a little bit to say it's about soil health and animal health, because they need to go together, and of course we have to link that to human health as well. And there's a lot more to grazing than just moving animals around. So sometimes when we think about grazing, we just think, oh, how many days are they in this paddock, or how many days are they in that one? And we're just looking at one very small part of a very complex picture. So let's just look at soil health just briefly, because, that's the thing that the ultimate aim is to do something to improve soil health. So, what is soil and by what processes does it form? We've just had this really very simplistic view for something like, well, probably since the Second World War, 70 years or so, of just looking at it just incredibly, just almost like one-dimensional. We've got the soil there, we've got plants that grow in the soil, we've got animals that are feeding on those plants, and we've just gone this really, really narrow-minded, if I don't mind me using that term, of, well, let's analyse the soil and see what's there, and if it's not showing on a soil test, which honestly is a very, very poor indicator of what you have in your soil, if it's showing there's not enough phosphorus or there's not enough nitrogen or something like that, then we'll just add that. So we're assuming that this is some kind of an inert medium, not a living system, and if it's short of something, we're going to add it. And then that has just produced so many unintended negative consequences that we really, really have to review that and start looking at soil in a very, very different way. In the same way, there are so many parallels with human health. For the last probably the same amount of time since the Second World War, you go to the doctor looks at the blood test, says, oh, you need to take this, and they want to prescribe something for you all the time. And sometimes that's become people's fault too. If you go to see your doctor and he doesn't give you a prescription for something, you think you paid your money for nothing. That people we always want to be taking something, or we want to be adding something to our soil, or giving things to our animals. So what is actually soil and how does this all work? Well, it's going to be weathered rock materials of some kind. It's going to be a combination of sand, silt and clay. And depending where you are in the world, it's going to be a very different combination. And as you all know, even on your own farm, there are very different combinations of those things depending on where you are in the landscape, down near the creek or up on the hill. But those weathered rock materials will not have formed soil unless they've been in contact with plant roots. So this is what I love about the presentation that we've just heard this morning. It's about using animal impact in that example to encourage plants to grow, because unless you've got plants growing and actively photosynthesising, you're never going to be able to do something about restoring the health of that soil. So plant roots are very important because plants, through that process of photosynthesis, are going to capture carbon dioxide, which is in the atmosphere at a very, very low concentration. And they're also going to take light energy, and they're going to encapsulate that light energy and that carbon from the carbon dioxide into sugars initially. And those sugars, now we've transferred light energy to biochemical energy, and that energy that's being captured in photosynthesis drives the whole soil ecosystem, because we have to start thinking about soil as a living system, and everything that lives in there needs energy. Just like we need energy, the soil needs energy, and the energy is going to come through photosynthesis and has to be modified by the plant. It's going to use some of that energy to grow itself, so it's going to produce cellulose, which is what the leaves are made from, and that's just joining sugars together to make longer compounds, more complex carbon compounds. It's going to make some lignified material for its shoots and stems and roots, and then it's going to produce, you know, flowers or seeds or whatever kind of plant it is that we're growing, and all of those things were made from sugar. So your soil is also going to be made from sugar, which is interesting, but sugar that is exuded by a plant, not when you add sugar. When you add sugar, it has the opposite effect. So it has to come out of plant roots, and this afternoon I'm going to be talking about carbon in soil and actually how that sugars that come out of the plant roots get changed into stable forms of carbon and how microbes do that. But these microbes have to be fed to do their jobs. And how do we know whether we've got exudates coming out of the roots of our plants? Well, you need a spade. Use a spade to dig up your plants and look at the roots rather than using a spade to collect a soil sample to send to the lab. We have to become better observers. In past times, farmers were incredibly good observers. We have taken away the responsibility. We've given the responsibility for observing what happens on our own farms. We've given it to an agronomist, in many cases, who comes, takes a soil sample, charges you for their time, charges you for the lab test, and then sends you a note to say apply this much P, this much N, whatever, and good luck. So we need to change that emphasis to you take responsibility, go out, have a look, see what your plants are doing, find out everything you can about how plants function, how plant communities function, and how your soil functions as a living system. So in this photograph you can see lots of soil sticking around the roots of that plant. And the soil is sticking around the plant because the microbes that are being fed by that plant are producing glues and gums, sticky substances to form what we call a rhiza sheath around that plant. And just about everything that happens in soil happens in that area. So the more plant roots we have, the deeper they are, and the more different kinds of plants we have that support different kinds of microbes, the more successful our soil is going to be as a living system. Just keep remembering, it's a living system. We have to activate it, we have to stimulate it, and we have to stop doing things that inactivate it. So if we take a close-up look of just what's happening inside that rhiza sheath, do I have a pointer on this, Gillian? Sometimes I press something and everything goes off, so I'm always a bit scared. This has got 6 buttons on it. And I only know front and back. One time before I pressed what I thought was the pointer and everything, that's it, that one. And everything went off and we couldn't get it back on again. So I'm a bit nervous now. Okay, so now we're looking inside a rhizo sheath. So here on the left-hand side is, that's where the plant root is. Here on the right-hand side, these are soil particles, and there's a gap between the rhiza sheath and the soil, the plant root itself. And inside that gap, there's all these are fungal filaments that we call hyphae of fungi, and there are hundreds of different species of fungi in there. Some of them are what we call symbiotic fungi, like mycorrhiza and trichoderma, those kinds of things, and some of them are just what we call saprotrophic fungi. They just like feeding on all those lovely sugars that are coming out of plant roots. And they're all highly beneficial in this case because we have colonies of bacteria in there we can't see at this level of magnification, and a lot of them are just living on the outsides of the fungal hyphae, because in the same way that the plant root is exuding all their sugars, they're then going into the hyphae of the fungi, and then they're leaking out of those a little bit too. So there's all these colonies of bacteria on the outsides of the fungal hyphae. Your fungi are really, really, really important in soils, and we have to, all of our management practices, we need to design them to have more beneficial fungi in our soil, because there's other things they do besides this. But one of the key things that happens in here is that a lot of those microbes, which will be bacteria and archaea, that are living on the outsides of those fungal hyphae are nitrogen fixers. So they're converting atmospheric nitrogen to a plant-available form of nitrogen, and they can't do that unless those fungal hyphae are there for them to live on, because the fungal hyphae are actually exuding energy. And in that very narrow space between the plant root and the rhiza sheath, you can get hold of a rhiza sheath on plant roots, and you can try this when you actually get to the point where your plants have got rhiza sheaths, and just very gently pull it, and the whole thing will just come off and expose the white root underneath it, and what you've got in your hand looks like a straw, like a straw that you could suck liquid through. So it's a column of soil around the outside of a plant root, and there's a very low partial pressure of oxygen inside there. It's not really an aerobic environment, it's nearly almost anaerobic, and that's really important because nitrogen-fixing bacteria are inhibited by oxygen. They're never going to be anywhere where they're exposed to oxygen. So when we see them on legume, when we look for the nodules on the roots of legumes, and those of you who were there at the field day yesterday would have seen some nodules on the cowpeas that were in the paddock. Well, inside a nodule, so the nodule has a membrane around it which stops oxygen from coming in basically, and it keeps all the moisture on the inside, and then you've got all this energy coming out of the plant roots into the nodule. So the environment inside a nodule on a legume is high moisture, low oxygen, high energy. If we can recreate that same kind of environment around plant roots, then we will have free-living nitrogen-fixing bacteria in there fixing all the nitrogen that that plant needs. So it doesn't have to be a legume. They're different kinds of bacteria. They're not the same ones that fix nitrogen in the nodule on a legume, but they fix nitrogen. Now think about this. A plant can't grow without nitrogen. It's impossible for a plant to grow without nitrogen. But if you look at all the greenery that's out there, I'll bet no one's putting nitrogen fertiliser on those trees. They might put some on that lawn maybe, but there's other plants growing under the trees, and I'll bet that lawn on the other side of the lake there is not being fertilised. And all the plants you see growing along the side of the road, I mean, just think about it when you're driving home today. Start looking around at all the plants that aren't being fertilised, and they're not legumes. The tree's not a legume. The things growing under that tree out there, not legumes. They can't grow without nitrogen. So we've got this fixation on legumes. Yes, legumes are a beneficial part of a pasture, but they're not the only thing that can fix nitrogen. But we have to graze our animals, manage our soils and our crops in a way that enables plants to form a relationship with free-living nitrogen-fixing bacteria. And a lot of them they can actually take up into the plant itself, and they can live within the plant. I haven't really got time to talk about that too much today. Well, I've only got up to about the third slide and I'm halfway. Never mind, keep talking, Christine. So fungal hyphae are also incredibly important for soil structure. So this is a photograph taken from sand, which they call soil in Western Australia. And you can see that the fungal hyphae are very sticky. They're producing lots of sticky substances, and they're actually holding those sand particles together to give it structure. Same thing happens in other soil types, but you'll have farmers in WA say you can't create soil structure, Christine. I'd say yes, you can with fungi. So what are you going to do to promote fungi in your soil? And when we have soil particles sticking together like that in these lumps that we call aggregates, then you have spaces between them. And the spaces between those aggregates are really important. Doesn't matter whether you've got a loamy soil or a heavy clay soil, like those of any of you who have heavy clay, you know how important soil structure is for your clay soils. You can't buy it in a bag, but you can get it with plant root exudates. And one of the reasons that we need to have well-structured soil is obviously for its water holding capacity and also for its ability not to get waterlogged when there's too much water. But also we need nitrogen. The atmosphere is 78% nitrogen. We need nitrogen to get down into these pore spaces in the soil so that our free-living nitrogen fixes that are living around the plant roots will be able to access it. So just in terms of what plant roots do for soil, this is a photo that was taken in Hungary on a soil that had probably been cultivated for something like 200 years or more, maybe even 500. And it was very, very compacted, didn't have any aggregates in it. And the farmer had planted a cover crop for the very first time. It was vetch and oats and Phacelia from memory, so I only had 3 plant families there, but that was sufficient diversity to cause some change. So we dug a hole in the soil and we could see it was very compacted. And this is just an example of what the compacted soil was like. The whole field was like that, and I'm sure any of you familiar with cropping have seen plenty of soil that looks like that. And this little oat plant had just completely changed. In fact, all 3 plants that were in all 3 species had completely changed the soil around their roots. So this is the impact that plants can have on soil structure, and it's obviously what you're aiming for with your mine site rehabilitation. So if we look at that just in a diagrammatic way, here on the left we have non-structured soil. All the soil particles just packed down, they're really close together, ends up being like a brick. And the best way to improve that is to get the soil particles actually have to stick together. So if they're together in a little lump like that, what we call an aggregate, then you've got space around it. So the same number of particles in that photo as in that one, but this one is a lovely porous, well aggregated soil. So these aggregates are actually engineering masterpieces. Mother Nature's been working on this for a few million years because it's very important for microbes to aggregate the soil. It's important for them to aggregate the soil so that they get all the things they need, like moisture, air that's 78% nitrogen, and their host, the plant that's feeding them, is going to grow better if the soil is aggregated around the plant. So it's in the microbe's best interest to have a healthy plant. And if we manage this whole system appropriately, it's a symbiotic relationship. It's how nature works. It's just like even when I was talking about the heavy metals, microbes will bind up heavy metals and take them out of the system, or other toxic chemicals and take them out of the system. They can do the same thing with salt, with sodium. They can do the same thing with aluminium if you have high aluminium soils, but they have to have the energy to do it. So these aggregates they actually are the fundamental unit of soil function, and that's what you want to see. Again, if you go out and you start observing and you're digging holes and looking at your soil, you want to look for that signs of exudation, and you also want to look for improvements in soil structure, which means it'll just be more crumbly and friable. But we have to have plants and microbes working together to produce these changes, and a lot of the things that we've done in the past have formed a disconnect between plants and microbes. So if you put on heavy rates of phosphorus fertiliser, for example, you'll cause a disconnect between the plant and the microbes. The plant doesn't have to feed the microbes anymore. You're just giving it phosphorus that it needs. Or heavy rates of nitrogen. Plant doesn't have to talk to microbes anymore because you've just given it the nitrogen it needs. You form a disconnect, you'll end up with compacted soil, even if you've never even driven on it. So we have to have bacteria, fungi and fine plant roots actually all working together. So there's a lot of communication goes on in the soil, a lot of interactions. There's a lot of things that we don't understand, and we're just starting to to get a view of what actually happens in soil. So in this diagram here, we have a fine feeder root. That the green coming down there is a plant root, and plant root hairs coming off the side. We often can't see those root hairs with the naked eye. And this blue circle around here is an aggregate that's forming around that plant root. And there's a lot of things going on, and there's a lot of information in that slide. That diagram is actually in an article that I wrote called Nitrogen, the Double-Edged Sword, which is on my website, and you can read a lot more about it in there. We don't have time to explain it much today, except to say that the little yellow ellipses in here are bacteria that are feeding off energy that's coming out of the plant. You can see fungal hyphae here. These are mycorrhizal fungi that are going right into the plant root and living inside the plant root. The fungi are pulling everything together to form that macro aggregate, but the little orange ones or red ones all through here, these are micro aggregates. So they're formed by bacteria. So we have to have bacteria forming micro aggregates, fungi forming macro aggregates, and they all need to work together. So you need lots and lots of different kinds of microbes in your soil, and the end result will be that instead of having a plant that's got white roots, you'll have a plant that looks something like that. And all of that soil structure is going to require root exudates, and it's only photosynthesis that can build soil. It is the only thing that can. So really our triangle, instead of having soil on the bottom and then plants and animals, which is how we see it when we look out in the paddock, we have to think all the time about photosynthesis. You always have to be thinking about, is this going to increase photosynthesis or reduce it? And if you use a refractometer to measure Brix levels in plants, you can see for yourself that you could have two plants growing side by side, take a Brix reading of the two, then just dissolve some urea in water and put it on one of the plants, measure the Brix levels half an hour later, will have gone down in the one that you put the urea on. You're, just shooting yourself in the foot using those kinds of things. You're reducing photosynthesis. Yes, you're feeding the plant, and yes, it will probably grow taller, but it won't have anything in it. And then your animal health suffers. So you've got twice as much plant because you put urea on it. It stopped building the soil, so you've now got compacted soil. You've got an unhealthy animal because it's eating a plant that's got none of the other things it needs. The animal needs calcium and magnesium and sulphur and copper and cobalt and everything from the soil. And if you've just caused a disconnect between the plant and the soil, you no longer have the pathway for all of those other things to come back to the plant, because not only is the plant feeding microbes, but the microbes are feeding the plant. So we don't want to cause that disconnect. So photosynthesis is at the base of our pyramid. This is the pyramid of life for all of us. We all depend on photosynthesis because we're either eating plants or plant foods like grains, or we're eating animals that have eaten plants. So we're not going to be healthy unless we do something about looking at this pyramid as well. So we've got green plants like carbon dioxide and water, the roots and the associated microbes, and only then can we form soil. So what we see in situations like, say, the southern half of Australia, where Grant's going to be speaking today, where he comes from, it's just been traditional for 50 years at least for people to have a crop that grows in winter when it rains sometimes, and then to have bare ground over summer where it doesn't rain very much. And we have to change that, because if you have bare ground every year for 50 years or something, you've only got photosynthesis. In fact, those crops that grow, they're only there for like 5 months of the year, not even there for 6 months really, once they get going, and then they get to a point where they're not exuding, not producing root exudates anymore. There might even only be 4 months where they're actually building soil. So that means you're just going to cut your soil in half. If you cut photosynthesis in half, you're going to cut your soil building in half. So photosynthesis, photo means light, synthesis putting together, that whole process is making life from light. And just to put that in a diagrammatic form, we've got carbon dioxide, energy in the light. We're going to combine them, make sugars, and these exudates in the soil are going to do a whole lot of other things in addition to what I just spoke about. But it will affect pH. You can see marked improvements in pH. If it's acid, it'll come back to neutral. If it's alkaline, it'll come back to neutral. It'll move back the other way. It's very interesting how microbes can juggle those things. nutrient acquisition is important. We want to have air in our soil, so we need porosity. We need porosity for the water as well. And then also the microbes themselves produce a whole lot of plant growth promoting hormones. The microbes actually produce hormones that help plants to grow faster and better. They produce chemicals, or biochemicals I should say, that protect plants from disease. And if you go back to the photos I showed of the rhiza sheaths, if you've got that thick rhiza sheath around a plant root, it's very, very hard for fungal pathogens or insects or anything to get in there and actually damage that plant. So optimising photosynthesis, which is really what we're talking about, requires grazing management, like good grazing management. We also have to be conscious of the plant growth stage. What kinds of plants are they? Are they annuals or perennials? Are they at a stage where they're actively producing exudates? What are the effects of plant diversity? And if we're going to take synthetic inorganic fertilisers, high analysis fertilisers out of the system, we need to be looking at some biostimulants and some biofertilisers when we're in the transitional stage, because we are a long way down now in terms of soil function. It's not working. In most cases, the soils are dead and inert. So we have to do something to stimulate those and just try and get that whole system working. So grazing management matters, and most of the people in the room here are pretty familiar with grazing management. Even though my talk is about grazing management, it's also about soil and animal health. So I'm not going to talk too much about just the grazing management, because there are often other things missing other than the management. And one of the things, like, it's not the cow, it's the how. We can't blame cattle for land degradation. It's how we've managed them. And one thing we need to be conscious of is what effect we're actually having on plant roots when we graze plants. And you'll all be very well aware of this, that, you know, if you graze a plant right down, like 90% of the leaf area taken in that example there, you're going to prune the roots, because roots are living systems as well. They depend on photosynthesis, and if we remove leaf, then we are going to prune roots into the soil. But sometimes that's beneficial. Sometimes you might want to have high animal impact and prune roots into the soil, because if this was our plant and we grazed it down to that, then all these roots down here have been pruned into the soil as organic matter. Now, that's not going to stay there as stable carbon, but in the short term, it's a bit like your dung and your urine, and you've got dung beetles, like taking dung down, and all those things. You're actually putting that labile carbon into the soil, which is a form of a biostimulant. It's not going to form stable carbon, but it's going to improve the soil underneath the plant, so the plant itself can grow better and photosynthesise more. Some people will aim to only take 30 to 50%. If you listen to Gabe Brown's videos, he'll be talking about, you know, never take more than 50%. And the reason for not taking more than 50% is if you take less than 50% out in a graze, you won't affect the roots, and sometimes you don't want to. So you need to juggle these things. Sometimes you will take a small amount out so that the roots, because we want to build soil down here, and plant roots can go, 2 metres. We've found them down at 6 metres in our soil cores. So sometimes you don't want to prune the roots. So you want to have a mixture of when you're thinking about your grazing management. As I said, we could talk about that for a long time, but I just want to mention that. The other thing is the growth stage of the plant. I think in the presentation this morning you talked about if the buffel grass was moribund and it's just sitting there, it's not doing anything, it's not building soil. If you grab it and pull it out, it comes out easily because the roots are not active. So what we see with grasses, if you look over the growth stages of grasses, or any plants really, but you're all more familiar with grasses, is that, it starts off, a perennial grass especially is a very small seedling, usually slow growing. Then we have these active growth stages, which normally you'd want to be grazing then when it's vegetative, to stop it from switching over to reproductive, because it's going to be much lower in nutrients when it's at this stage. But if we're looking at root exudates, we find that it's in this actively growing period of plants where they produce the most exudates, because they need to feed the soil to get the things from the soil that they require to grow, especially nitrogen. When they get to the point of actually producing seeds, they remobilise nutrients from their leaves and put those into the seed. They don't get them from the soil at that stage. So whatever they've got in the plant, when they get to the end of the vegetative stage, that's what's going to be mobilised into the seed. And that's why, from a grazing perspective, once a plant has made that decision to switch over from vegetative to reproductive and starts mobilising things within the plant, that's why its value for forage falls, because it's putting stuff into the seed and you're going to have lower protein and everything in your plant. The other thing is looking at the difference between annuals and perennials and just how quickly these exudates do fall off. So there's a lot of information on this graph, I'm sorry, but what we have here is how much of the carbon, so this is the below ground allocation of carbon in the form of root exudates that a plant put into the soil. So a plant can photosynthesise, could possibly use half of the sugars to make itself, to grow leaves, and half of the sugars to feed the microbes. That's a sort of a pretty normal sort of a situation. But when the plant is very young, it actually allocates a lot more to the below ground, because that's when it has to establish and form relationships with soil microbes. As it gets more mature, and especially when it switches over to reproductive, it doesn't care too much about soil anymore, doesn't need it anymore. I'm just going to produce some seeds, and if it's an annual, then it's going to die. So these purple ones here are annuals, and you can see how early, this is plant age, days after planting, so 100 days, like say 3 months. Within 3 months, they're pretty much done in terms of producing exudates. So if you really wanted to build soil, degraded soil really quickly, and you could get them to grow, and you had the money to spend on seed, you get a whole heap of annuals in there, and you need lots and lots of different kinds. I don't know how much time I'm going to have to speak about diversity, but I think I've got a few more minutes to actually, because the more different kinds you have, the more exudates they will produce. Perennials, like most of your grasses up here that are perennial, your rhodes, your signal, all those kinds of plants do keep on producing exudates, because, I mean, some of those perennial grasses can live for 50 years and 100 years or something, so they're going to just keep on producing exudates. But it would look from this graph like perennials produce more than annuals. I've heard Joel say it, and you've been working with Joel Grant, and I heard you say it yesterday too. They produce more as a function of how much photosynthesis if they produce a certain amount in photosynthesis, they allocate more of that to the soil, but they don't photosynthesise as fast as annuals. So even though annuals give less of it to the soil, they photosynthesise much faster, because an annual's got to get up, grow, and be done, and produce some seed. So it's going to do everything it can. So your annuals are actually going to build soil faster. Perennials are there for the longer term. And it's not enough just to have green plants. Diversity really matters. I cannot emphasise this enough. You cannot build your soil with just grass. You cannot build your soil with just grass and legumes. You can build it a little bit, but you really need at least 4 plant families there. And we talk more about plant families a little bit later, but activating the soil microbiome with plant diversity has been the thing that in the last 10 years especially, probably 20 years, but in the last 10 years has been the practice that's been adopted the most widely around the world in everything from vegetable farms to agroforestry to like cropping, grazing, everything, and just about everyone who's actually embraced it and embraced it correctly. It's like grazing management. You've actually got to understand what you're doing, and you need your 4 plant families there, have seen such phenomenal results that they probably would never, ever go back to having a monoculture of anything. And you could have 10 different kinds of grasses in there, you've still got a monoculture of grass. You've only got 1 plant family. So biodiversity is the key factor in the effective functioning of every living system, including your gut. The human gut requires microbial diversity to function effectively. The rumen requires microbial diversity to function effectively, and soil requires microbial diversity to function effectively. So soil microbial diversity is so important for the structure, for nutrient cycling, for resistance to pests and diseases, for your productivity, And then if we're just talking of global systems, you know, for food security for everybody really, or even in just regional food security, and for landscape function and carbon sequestration, which I'll talk about this afternoon. I mean, basically everything. Soil microbial diversity is absolutely fundamental to everything that happens in your soil and everything that happens on your farm. And if we look at natural grasslands, I know this wasn't a natural grassland here, but if we did have a lot of natural grasslands in Australia, and we had at least 500 to 700 species. I've seen some of the prairies in the United States where they've had even more than that. They've counted 2,000 species. It's hard to imagine, and they're all edible. And our species were all edible too. Now, was it last year or the year before? I can't remember now. I was at, is it called Basalt River Station, just north of Collinsville, and we were out just looking in the pasture, and we found literally like at least 100 little plants that were only about this high that no one knew the names of. We had some botanists there with us. They didn't know what they were. I had no idea what they were. But they were little native plants that grow in grasslands, and we're not talking about necessarily a big bulk of feed. When we're talking about diversity, we're talking about having different plant families there, because everything that happens underground is going to affect the nutritional content of our grasses. So when you look across it, you'll just see grasses, which is why we called it a grassland, because that's what we see. They're tall, sit above everything else. But if it's really healthy, you get in and look through all those grasses, you will find, as we found there, hundreds of things that nobody even knew what they were, but we could see that they were different and they weren't grasses. So we knew we had diversity, and that's something we really need to hone in on and find out what those plants were. So we can recreate that diversity. We've lost it, nearly all of our land, or if it was a forest here and it's now you want a grassland, you can recreate that diversity to some extent with our what has become so popular cover crops, multi-species forages, multi-species everything. And the thing I see that's really missing from here, from these landscapes, our grazed landscapes, are our deep-rooted forbs. So when you think diversity, just stop thinking about grasses, please. You could have an entire forage, perennial forage that didn't have any grasses in it, and you'd probably be better off, but you can't keep them out, they'll come back. I mean, the reason we have grasses is they're survivors. They're absolutely amazing. It's almost impossible to keep grass out of anything, so why would you spend money planting grasses? So we need some of these things with deep roots. One of my favourites is chicory. I don't know whether it grows here. I've seen beautiful chicory up on the Atherton tableland, so it's not to do with, you know, how close to the equator you are, but there could be other factors here that maybe it doesn't like humidity. I don't know. But there are all these deep-rooted forbs that are going to do so much to change soil that we need to figure out actually how to grow them. I think I'm up for my time. Oh, she's going to give me 2 minutes. Oh. Okay, so there was this biodiversity experiment that ran in Germany for 15 years. In fact, it's still going, I'm pretty sure. And they had 1, 2, 4, 8 or 16 plant species from 4 functional groups. So if they had 16 plant species, it wasn't 16 grasses, or 8 grasses and 8 legumes. So they had grasses and legumes were in there, yes, but they had tall herbs, so something like sunflower would be a tall herb. A herb is just something that's not a grass, doesn't have to be parsley. And a short herb is something like plantain or whatever and that was how they broke it up. But all we need to think about is if you have 4 plant families at least, you will have tall herbs and short herbs in there. And they looked at a whole lot of stuff, biomass and insects and soil microbes and water balance and, you know, carbon, nitrogen, phosphorus. This is what it looked like, an aerial view of this site. So where you see all one colour, that will be where there was just one thing planted as a monoculture, and then all the motley ones where it's all mixed up. So as they put more plants into their mixes, if they went from 1 to 2 to 4 to 16, biomass increased. This is what we call over-yielding. Everybody everywhere in the world talks about this. Over-yielding happens. So you could have something like we talked yesterday about, you could have corn grown on its own, faba beans grown on their own. You probably wouldn't even think of putting corn and faba beans together. There's a lot of benefits to putting a warm season grass and a cool season legume together, surprisingly. So a lot of cool season things that wouldn't normally grow in the warm season will grow in the warm season when you put them with a warm season grass. It's quite amazing. But anyway, we put this shows you that as the number of species, which is along the bottom here, 2, 4, 8 and 16, remember they're from 4 plant families, as we have more plant species, we get more biomass. And not only that, so it's not like we produce more stuff that had less in it, it was also more nutrient dense, and it had more beneficial insects in it, and it built soil faster, and the plants exuded more carbon into the soil. All of these things, absolutely everything, not just biomass increased. So it's not like we say something like buffel grass where you grew more buffel grass, but it may not necessarily be better, although in this case it was. Everything improved with the number of species, the number of plant families in the mix. I'm just going to skip over these and just show you what happened with the soil. So here we have a 2-family mix, like say grasses and legumes, for example. You can only ever build a certain amount of soil. Yes, I'm just getting that one right. But where they had at least 8 different species in the mix, they built deeper soil. All right, so I'm going to stop there and just have some Q&A on that, and if there aren't any questions, I will keep going. And the slides that I haven't covered, I'm not sure what number I'm up to, but I know I'm not very far into the presentation, but I'm going to put some of these, what I haven't covered, I will put into this afternoon's presentation and make sure I do cover everything there.
Questions? Don't be shy.
Yeah, Christine.
Whoop. Can we get a microphone?
I don't need it. Christine, we have a paddock where it's just, well, there's only really one grass in it. How would you suggest, or what would you suggest for us to be able to get more diversity in that paddock? Like we graze quite well. I know we graze quite well, but we're just not getting the diversity in there. How would you suggest to get more?
Okay, so one of the things I was going to show you, I'm not sure whether it was in this presentation or this afternoon, but I will show you, is that as most of you are aware, federal government introduced a soil carbon scheme for like rewarding farmers for increasing soil carbon. And the first farmer that was successful in building their carbon, I'm actually going to show you some slides of how much they've increased it by, a phenomenal amount, almost an unbelievable amount, but it was measured under a government scheme using all of their protocols, which took them about 10 years to come up with the protocols. So we have to assume that the measurements were correct. And this was Niels Olsen in Victoria, and he uses a machine that he developed himself. So this is not necessarily a promotion for his machine, but he has built soil carbon. It's called a Soil Key Renovator. I'm going to show you a photograph of that. What it is that it's strip tillage. So it's quite vigorously, it's got a little strip tiller on it that quite vigorously cultivates the soil in a narrow path, but like right across the whole thing, obviously. So it cultivates the soil where you're going to plant the seeds, and it leaves a gap, then it cultivates the soil, leaves a gap. That's that kind of a thing. And so you can leave some of your pasture there, but you're going to cultivate the other, so that you can actually get seed establishment into those grasses. You're not using a herbicide. The cultivation is very shallow. So there's been concerns about cultivation. Moldboard ploughing is a problem, right? You're going to completely invert the soil. And there are many forms of tillage that are a problem. But there is another thing that came up yesterday that I was going to talk to Grant about. We ended up not coming back in the same car, so we didn't. And that was about mycorrhizal fungi. We got this incredible fear of tillage damaging mycorrhizal fungi. Now, in the ways that tillage was used in the southern half of Australia, where you had a fallow over summer, and in many parts of the Downs up here too, I think people have bare fallows, and if it does rain and you get the green tinge on the bare fallow, which is some kind of weed that's decided wants to grow there, people will either spray it or in the past would till it. So the soil was left bare. If anything green grew there, it was cultivated. And there was a lot of high analysis fertilisers used in those soils, a lot of herbicides used in those soils, and a lot of tillage used in those soils. Tillage got blamed for reducing the counts of mycorrhizal fungi, when in actual fact was a combination of all those things. Mycorrhizal fungi, I showed you those photos, right, of the fungal hyphae that are coming out. If you have shallow tillage, what I mean is like nothing more than that much. Or even if you have tillage in a band, even if it was deep, but it was only in a narrow part, so you're not tilling all of the soil, you're not turning it all over. If you actually break those mycorrhizal hyphae up and then plant something, they will reconnect with what you plant. So it is actually easier to establish mycorrhizal fungi from hyphal fragments than it is from fungal spores. And our soils are adapted over millions of years to disturbance. You have bandicoots and things like that here that dig around in the soil, and where they dig, it actually becomes more productive because they disturb it and they break up the hyphae, and those provided plants then grow there. So the problem with tillage is we've disturbed soil and left it. And then if plants grow, we come and till it. In the past, you'd have 6 passes or something before you planted a crop, and you'd work it down to a fine till. That's over-tilling. So when just looking at the grass yesterday, the signal grass, I was thinking if you want to establish 4 plant families in here, you're going to have to do something with that signal grass. You don't necessarily want to kill it all. You don't want to spray it out, probably. But it's not going to hurt you to have shallow tillage in there, and something like the soil key just tills some of it, even if you don't want to, till all of it.
What do you call it in soil key?
It's SOIL, soil, and then all one word KEE. I'm not sure where they came up with that. If you Google soil key renovator, you'll find a lot about it. But I would definitely I'm not in favour of herbicides, because the more we see, Grant talked a lot about this yesterday, the more we see with so-called safe herbicides, I'm not even talking about human health here, I'm talking about plant health. So I spent a lot of time working in the United States. Human health is an issue with herbicides. Anything ending in "side" kills things, and they're all problems. But in the United States, as you're all aware, they have genetically modified corn and genetically modified soy. I think 90% of their crops are genetically modified, also their potatoes, their apples, it's like you get, what can I eat? But if you go into a field there that's just been sprayed with, corn or soybeans that have just been sprayed with Roundup Ready soybeans, Roundup Ready corn, it looks terrible. The plants all go yellow. I mean, they're not really, they can survive being sprayed with Roundup, but it does incredible damage. The soil looks terrible. The whole system looks so sick that they're not really Roundup ready. and Grant was talking yesterday about just like any post-emergent or even some of your pre-emergents, you just don't realise, unless you leave a strip or you accidentally leave a strip, sometimes the sprayer stops working or something, and we get shown, hey, this is what the plants would have looked like if we hadn't sprayed them. So I'm not keen on herbicides. I have no problems with soil disturbance, provided it's limited and you know what you're doing. And don't worry about breaking up mycorrhizal fungi. If you've got something living there, it's going to be full of mycorrhiza, and if you put something else in there, you're going to disturb the soil and plant something. So I can see lots of reasons why the soil key works, but it works. And he has built massive amounts of soil carbon. It's just incredible when you look at the figures. And every year, like he built a whole lot, and then the next year in 12 months, he built more than even I, who've worked on soil carbon for a long time, could believe, but it's been built under it's been measured under a government regulated scheme. He had to be able to do it. And then this year again, he has increased his soil carbon again. I'm talking like 6 tonnes per hectare or something of carbon. He's sequestered like 12 tonnes of CO2 per hectare per year. In 2 years, he sequestered nearly 50 tonnes of carbon dioxide. He pulled 50 tonnes of carbon dioxide out of the atmosphere per hectare on his farm and stabilised it in his soil with plant diversity. Oh, that's what I didn't tell you. He uses the soil key to plant forbs, chicory, plantain, lucerne, like whatever. And they're all, okay, things that grow in Victoria. And he has done it. Remember that diagram I showed? He's done it actually. There's a good thing here. If you have these deeper rooted plants here, that if you want to, and here's the other thing, I was going to talk about this afternoon, we'll do it now. If we drew a line across here, if we said, let's just say that's the top 30 centimetres of the soil there, your top 12 inches of your soil. So under the federal government soil carbon scheme, using their protocols, you use a hydraulic soil corer, you measure all the way down to 1 metre, how much a metre is, and it's divided into 2 sections. It's just the top 30 centimetres, and then from 30 down to 100. There's just those 2 sections they look at. They're trying to do it like as cheaply as they can, but it's very important that they measure down to a metre. So with Neil's, he's got these deep-rooted things like chicory and lucerne and whatever in his mixes, and plantain also has very deep roots. Over the last 3 years where he's taken, in 2 years where he took 50 tonnes of CO2 out of the atmosphere and stored it in his soil, no change in the top 30 centimetres. So if you're only taking a 10 centimetre soil sample, you don't find anything. It's just labile carbon up there. The change has all been below 30. So it's been in that, from 30 to 100 is where he has stacked all that carbon. It is so safe down there. Like it can't be, it can't be denatured, it can't be oxidised, microbes can't break it down. I mean, he has stacked, stored, sequestered, whatever word you want to use, stable soil carbon, which I'll talk this afternoon about how that actually happens down there at depth. But you can't do it unless you've got plant roots down there. And the more different kinds of plants you put together, the deeper the roots go as well. So even something like lucerne or chicory that has deep roots, when we put them together, the roots are even deeper. When we put them together, they exude more carbon than they do when they're on their own. So one of the things I was going to tell you today is that we can't tell from a monoculture what plants are going to do in a polyculture. And that is something that has become increasingly obvious over time. And also that plants that you think won't grow in an area or you think won't grow at a certain time of year will grow when you put them with other plants that are very different to them. So maybe, Gilly, I might just, we've got another question, can I just pop over to something? Well, I've got the microphone, right? Okay. So I was just going to talk about soil structure. I'm not going to go into that. But what happens when it gets wet like this is that this is the same experiment. This is the Jena experiment in Germany. That was underwater for 3 weeks, waist-height water. They thought, that's it. 15 years, it's all gone down the drain. We've lost everything. Guess what survived? All their multi-species plots. What water? They lost nothing in their multi-species plots. They lost all their monocultures. Okay, so if we have different kinds of plants growing together, in this example here we just have a bean, there's just a soybean plant and a rice plant, but they have very different microbiomes. When we have very different microbiomes, because plants support different species produce different families, I have to stop using that word species, different plant families support different kinds of microbes, they actually interact and then everything changes. It just changes all the rules. So if they're all the same, if we just got all grasses, they compete with each other. They have and it's been shown, it's been proved in science, there's negative feedback. They're not just competing for water or nutrients or anything, they actually have a negative feedback effect on each other biochemically. The microbes have a negative effect on each other, and that affects everything. How much nutrient they can take up, how resistant they are to pests and diseases and all those kinds of things. If they have very dissimilar microbiomes, you want to put really different kinds of plants in there. It has a positive feedback on all of those things. But this is just one little slide I wanted to show you, that it's not possible to tell from a monoculture how plants perform in polyculture. And this photo was taken accidentally. This is from Chase Thornhill, who's the manager of the Mendocino Wine Company in California. He bought 12 species of cover crop seed from green cover in the United States to put in the interrows of his vineyard because he's heard that cover crops are a good thing to put in vineyards and it's good to have a lot of different kinds of plants there. But he was he's a grape grower. He knew nothing about cover crops. He knew nothing. He didn't know what all these different things look like. So for his own benefit, he went to the hardware store, bought 12 little plant pots and some potting mix in a bag, and he planted each of the seeds into these pots. So he's got, maybe peas and oats and barley or whatever, all the different species, and so that he could identify them. So he thought, when I plant my cover crop and I'm out there in the vineyard and I'm looking at it, I know what peas look like and I know what barley looks like, and I know what whatever else he had in there, faba beans. I can see there's faba beans in there. And then he thought, well, I wonder what would happen if I just put them all together. So he put all 12 seeds into this pot. Well, there's chickpeas in there, I can see. There is absolutely no fertiliser. There is absolutely no relationship between how the plants are growing in there and how they're growing here on their own. Look how nutrient deficient and everything they are. So the thing is that we plant a monoculture and it comes up this colour. You go, oh, we need to apply some nitrogen, and probably do in a monoculture, because how is a monoculture going to get its own nitrogen? It can't. Because you need diversity for all those things. And even though he just did this for his own purposes, it just blew me away when I saw that photograph. That's what happens when you put all these different kinds of plants together. So here's another thing. Some of these, like this one here, these are legumes. They're supposed to be able to fix their own nitrogen, but they're looking very nitrogen deficient. So even though they are supposed to be able to form a relationship with nitrogen-fixing bacteria, it's very difficult for them to do it in a monoculture. So that really kind of blew me away. Oh, and I will just say one more thing. Yesterday I told her I was going to talk for half an hour, and about 3 hours later she kind of just dragged me off or switched off my microphone or did something.
Kicking and screaming.
And I've got the microphone, and I'm higher than you. I'm here. So, apart from all the science, and we have so much science around this now, it's really exciting. But on-farm experience has shown that diverse forages can not only replace, but they will outperform low diversity swards plus all the fertiliser that you want to put on them. But they have to be diverse, and they have to have a minimum of 4 plant families. So I'll just leave a couple more questions, minutes for questions. I'll be good.
So we have 3 minutes for one more very quick question.
Hi, Christine. I loved the photo of the different pots that would make a great kids' classroom experiment.
Yeah, I'll put that one back.
Yeah, that's
that
brilliant.
blew me away when I saw that. And he didn't, he just wanted it. He wanted to know what they looked like.
I'm going to try that.
Yes.
I'm going to try that for curiosity. So you're talking about plant families. Is there a resource, website, book or something? Where can we learn what the different plant families are and what plants are within those different families? So we can
Great
make
question.
Sure we get 4 different
That came up yesterday, and I said to Gilly I would send her a list of the 17 plant families that are most commonly used in gardens and agricultural and horticultural situations like this. But you can also find out really easily for yourself if you were wondering about a plant. So if you were wondering about sunflowers, for example, just put into Google, what plant family are sunflowers in? And it'll come up, it'll say probably the daisy family, but then it'll give you the name of that family, which is Asteraceae. So grasses are, in their grass family, and that's Poaceae. Legumes are in the Fabaceae family. All the daisies are in the Asteraceae family. So you just make sure that you have your 4 plant families. So some of the common things that people use in cover crops, chicory is in the daisy family. It's an Asteraceae. It's in the same family as sunflowers. Doesn't look like a sunflower, but it is in that same family, highly mycorrhizal. If we look at the southern half of Australia, the First Nation people cultivated yam daisies really widely. Some of the first Europeans that went there talked about just fields of yellow. Yam daisy is very, very closely related to chicory. So it has a taproot, which is what Aboriginal people used as a staple, was their staple carbohydrate source. And it is in the Asteraceae. And chicory is really closely related to yam daisies. It's just got a blue flower instead of a yellow one. Another widely used thing that's in that same family is yarrow, but I don't know whether it will grow here, has very deep taproot. And then down south we can grow things like, we can grow buckwheat, which is in the Polygonaceae family, so very, very different to grasses, very different to legumes. It may not grow as a monoculture here, but you should try it in a polyculture. As I said, everything changes. See whether it will grow, probably in winter time. Down south it gets frosted in winter, but if you don't have frost in winter, it won't like the heat probably. I'm not sure. Even if it grows a little bit, I said, and remember that plants in the early stages, that's when they produce the exudates. If your buckwheat only grows this big but it's in there, it's going to be doing something great for mobilising phosphorus. Best thing on the planet for mobilising phosphorus in your soil. And then there's Phacelia, which doesn't like tropics, but that's in Boraginaceae. That's widely used down south, probably won't grow so well here. And then there's a burnet, which is in the Rosaceae family. So it's in the same family as strawberries. It actually looks a bit like strawberries. Again, I'm not sure whether burnet would grow here, but maybe in a polyculture it would. And then you've got your plantain, which is in Plantaginaceae. So I will send that information to Gilly, and I'll get off the stage before I lose my life. She's so mean. She was so mean to me yesterday.
I'm so glad.
You can tell, can't you, just looking at her?
I'm just glad it's Gilly who's copying it and not me.
So, so what I didn't cover today, this morning, I will pop into my presentation this afternoon. But I think that's a great take-home message, and how interesting that it was just put together by someone who just wanted to know. And this is where a lot of our answers have come from, someone who just tries something. And you won't necessarily find all the answers on the internet. Yes, you can find what families plants are in. You won't necessarily find whether those plants can grow here or not. They might not grow in monoculture, but they might grow in polyculture. And people are starting to put very strange things together that you wouldn't think, and it just changes everything.
Please thank Dr. Christine Jones. She will return this afternoon with another presentation, so don't worry. There's plenty more information to come. I do